Hello and welcome to the Beyond Biotech podcast number 102. I'm Jim Cornell and this is the weekly podcast from Labiotech. It's a little bit different this week in that there are three guests all covering the same subject, which is radiopharmaceuticals. It's a hot topic currently, and so who better to talk about the subject than Ken Herrmann, a radiopharmaceutical academic expert, and he is Professor of Nuclear Medicine at the Universitätsklinikum Essen in Essen, Germany. And there are two people from the Swiss biotech company Molecular Partners, and they are Patrick Amstutz, who is a co-founder and the CEO, and Dani Steiner, SVP of R&D. In our wide-ranging conversation, you will hear first from Patrick Amstutz, then Ken Herrmann, and last but not least, Dani Steiner. I wonder if, to get things started, I could get a little bit of background on Molecular Partners.
Sure. I think I can take that one as a co-founder of the company. So I think there's two things. One is Molecular Partners and the other is our DARPin technology or DARPin modality. And now actually, I think I will start with the DARPin modality because the company was founded because of the opportunity of the modality. And there is some logic to it. So in the late 90s, we were working in the group of Andreas Plückdun, an antibody engineer. And we saw limitations of antibodies. And antibodies are really great molecules. They bind their targets specifically. They have unique stability and a lot of functions. But they're very large and complex. So one antibody is 4 proteins, actually, in 12 domains. And we were interested in what had nature evolved next to antibodies as binding domains. And we found a whole class of proteins called repeat proteins. And that goes back to the genome sequencing efforts when one could, back then, really mine those genomes. And that's where we found ankyrin repeat proteins, leucine-rich repeat proteins, and also other repeat proteins. And we chose the ankyrin repeat protein and built the designed ankyrin repeat protein, in short, the DARPin. And the DARPin is the 10th the size of an antibody, small, stable, one protein, one domain, and it shares the high specificity and affinity of the antibody. And so with that, the idea was born to create a novel therapeutic modality. So we were not interested more in, call it diagnostics or tools, but we said, let's really use this novel modality to make therapies. And we always had two ways forward. And the idea was always not to be an antibody because antibodies are great. So let's do things others don't. And one is super small, simple, high affinity binding in one domain. And the other is multi-specificity. I mean, just we were joking before about the football cup. That's the team. So build a team of domains to fight the disease. And we have ventured into both, and that's where Molecular Partners comes in. So we have built a team of experts around the idea that, and our slogan is make drugs that matter. So use the DARPin modality to make drugs that help patients, so high medical need, ideally single agent active. And so far we have brought 7 DARPin candidates to the clinic. We have treated more than 2,500 patients, and we're really excited now to be in the radiotherapy space with support of Ken, who is also here on the call.
I wonder if you could give me a bit more background on radiotherapeutics and radiopharma, how it actually works and what kind of diseases it's suitable to tackling.
Jim is a perfect assist for me. So I'm a nuclear medicine physician. And the truth is that in nuclear medicine, we use radioactive substances for imaging as well as for therapy for many, many years. And indeed, the concept of using one radioactive substance, iodine, for diagnostic imaging, but also therapy of both thyroid cancer, but also benign thyroid disease, dates back to the 1940s. Saul Hertz was the person who first implemented this. Now, this being said, both radioactivity and also nuclear medicine doesn't sound very, let's say, fashionable. And this, I think, is what happened in the early 2000s when people were looking for a new term and this new theranostics, radiotheranostics came up. The idea behind is the same. We want to target one specific structure and deliver the radioactivity, the radioactive radiation very precisely to this very dedicated structure. And as an example, and this is one of the most known one, is actually prostate cancer. And in prostate cancer, such a target, which is highly expressed in prostate cancer and almost not at all in other healthy tissues, is the prostate-specific membrane antigen, PSMA. And for this target, they have now discovered a ligand which binds with a very high affinity to this target. And all we have to do then is to use this ligand, make it radioactively hot. We can use diagnostic radionuclides for doing imaging or therapeutic ones for therapy. And this is actually the big breakthrough. For full disclosure, between thyroid, 1940, and Pluvicto, which is a medication for prostate cancer in 2022, there are also a couple of other therapies in between, but not many. But just to mention very quickly, Lutathera, targeting somatostatin 2 receptor in neuroendocrine tumors, and also radium-223, targeting bone metastasis, especially in prostate cancer. And your question was, what are the tumors for the future? I think it's something that you're going to discuss in the next minutes about. But of course, the big aim is now, after neuroendocrine tumors and prostate cancer, to also advance into other very important solid tumors. And my dream there is, of course, GI tumors, lung cancer, and breast cancer.
What are some of the challenges that you face or some of the obstacles for radiopharma innovation?
So now, being the longest in the field, I will take this one as well. A big problem is obviously the commercial success. So around 2000, there were two really interesting antibodies for lymphoma, different kinds of lymphoma. And even so that they were medically very successful, they were never commercially successful. And the reasons of them are multifold, but one of them, first of all, it's important that a therapy which includes radioactivity has a certain shelf life. So we cannot just store it on the bench and then keep it and eventually use it. Once we use it, they decay, they lose activity over time. They're logistically a little bit more complex. And then the third, and I think the most important part, is that for a very long time, we also were not able to gather enough convincing data. So when you go nowadays into a tumor board and you want to actually convince our clinical fellows to send you a patient, you need to deliver prospective phase 3 data. And the truth is that for a very long field, nuclear medicine and the diagnostic community did not succeed in performing this prospective phase 3 data. And I think this is one of the biggest consequences of the recent successes now, that we see more and more people who really have a lot of experience in drug development entering this space and moving forward. And this is maybe also something where I would like to directly hand back to our friends from Molecular Partners, how they see the space and how they are going to help me and my colleagues to actually overcome the hurdles, which I just mentioned.
Ken, I will hand over to Dani for more the technical parts. But one point I found interesting, because 2007, we first time looked into this. And what Ken was mentioning before with the supply chain, that you would sort of deliver overnight, 2007, that was just, let's say, a dream. And I do think over the last 10 years, including COVID, where this Amazon, I order one day, the next day I get it delivered. I think this has really changed how we think about supply chains in general, not only for radiotherapy. And then the other one, the CAR T space, where you make an individualized product for a single patient. So those two things, the idea of you can actually manufacture for a single patient, and you have an ability to deliver whatever you want, the book or a part of your bicycle overnight, together really opened the space for radiotherapy. So I think those are, for me, sort of the simple general things where society has a different expectation. And I'll now hand over to Dani to go now more into the technical parts. What does radiotherapy need? What are we solving for? And what are the now more technical obstacles?
Happy to take that question. And maybe I start from where Ken already nicely summarized. So the overarching ambition is, of course, to now translate or expand that great success that has been seen for PSMA or somatostatin receptors into as many as possible other tumor indications. And starting there from the underlying requirement, you need, of course, the radioactive part, but on the other side, you need targeting moieties that address the requirements of delivering the radioactivity very specifically to the tumor, while sparing the dose-limiting critical organs, which are on one side the kidney very often, and on the other side the bone marrow, leading to bone marrow toxicity. And that's whenever you have targets which have like pockets where you can identify such small molecular weight ligands like PSMA or somatostatin receptor, there is like you go with these low molecular weight ligands, they're perfect. They're quickly into the system, they quickly leave the system again, accumulate high on the tumor, and then ideally stay long enough to deposit the energy on the tumor. But the problem is this space of targets or the number of targets that can be addressed this way is very limited. There is only a few of these targets available. And that's why people have been looking into expanding this using more classical, I say, targeting moieties to expand that space, using on one side antibodies, small proteins like DARPins, or then peptides and cyclic peptides. However, the problem is all of them bring limitations. The antibodies, very long duration in circulation, leading to a lot of decay of the radioactivity in circulation, and often also limited by tumor penetration. Still, some companies and teams are following this avenue. Then cyclic peptides as well, leading to often high kidney accumulation and also challenging to generate against certain targets. And then the small proteins where you see on one side, they lead to high kidney accumulation as well and have somehow mediocre, I say, tumor accumulation. And that's exactly where we've been seeing the opportunity for DARPins to figure out solutions that would overcome these limitations.
Has there been a shortage of radioligands, and has that been the challenge?
That's a very important point, Jim. You mentioned that the big challenge has been for a certain time the access to radionuclides. There are a couple of reasons, and this has been not only for the therapeutic ones, also for diagnostic ones, because some of the reactors are very old, then they needed to be undergoing maintenance. Then some of them were shut down because of unexpected need of maintenance. This is one of the things which people always think about when they talk about supply chain shortages. The second one, of course, is a big issue Novartis as a frontrunner eventually had when one of their production sites was actually shut down and the other one was not opened yet. And to be fully honest, this is the growing pains which a field has. And this was very nice outlined by Patch as well as by Dani. But I think we have overcome these growing pains now. We see that Novartis as a frontrunner has really matured. They now have a couple of production sites. So I think for the future, the access to Pluvicto and Lutathera of the approved products is figured out. We also see a lot of investments into different layers of supply chain, which means on one side, the production of the radionuclides. I mentioned very quickly here, actinium, lead, also terbium. But we also see a lot of investment into labeling capacity, so-called CDMOs, contract manufacturing. And we also see a lot of investment actually also into companies who really specialize in the logistics. So yes, it's a very well-taken point. There have been significant challenges in the field, but I think overall in the last 5 years, the field of theranostics has matured significantly and will continue to mature over the next development cycles.
And how have you kind of taken that on at Molecular Partners, I guess, with your own technology?
I can just give sort of the intro and then hand over to Dani for the technical, call it solutions. But we actually had a very successful COVID program with Novartis 3 years ago. And Novartis told us, hey, your small proteins would be ideal vectors for this approach. And so we looked at it and then we said, okay, what do we need to solve? We need to get high affinity specificity, we have that. Get a lot on the tumor and little in the kidney. We don't have that yet. So we need to work on those 2 dimensions. And in the last 3 years, that's what the team with Dani has done. And maybe you can give us some background on how we actually solved first, maybe the kidney accumulation, so no kidney accumulation. And then second, how we bring more DARPin into the tumor.
Yeah, happy to do so. So that's, as I mentioned, that's like for any of the small proteins, which you can use to target a very broad class of tumor antigens, of tumor targets, you have these 2 same overlying challenges to address. And that's where we've seen, looking at these challenges and understanding the properties of the DARPins very well, that we felt like from the start, there would be good ways how to address this. So the first one, the kidney, that's where we used an approach, and maybe I need to quickly go a bit into the background for those who are not so well aware of the scientific aspects of this field, is like the kidney basically, whenever you have a small protein or a peptide that is excreted into the primary urine, should go into the urine, the kidney has established a very efficient mechanism to reabsorb that protein source, to not lose that protein source or nitrogen source from the body. And that's where we realized that reabsorption mechanism by a heavy re-engineering of the surface of the DARPin, which is enabled by its very high stability, would lead to what we call a stealth DARPin, that is, instead of being reabsorbed in the kidney, going directly into the urine and can be excreted by the individual. So that was the first avenue to solve the kidney aspect. And the second avenue to solve the tumor uptake aspect, it's on one side, we knew since a long time that high affinity helps to get high tumor uptake, but we realized that on its own is not really sufficient enough to get good enough loading of the tumor, enough energy onto the tumor to get an effective tumor kill. And that's where we started to build a platform for slightly increasing the systemic circulation time of DARPins in order to get more loading onto the tumor. We've heavily invested to get this done in a way that the blood levels are not strongly going up, but the tumor is really driven up to high levels. And this is the basis which we established and also enabled now our first molecule in the context of DLL3 as a target and lead-212 as a radioactive payload to be moved into the clinic. And I think really that this MPO-712 is on one side, it's of course a great drug moving forward, and on the other side, it's unlocking to do many more targets now in the same context.
I guess recently you were at the SNMMI conference. I wonder if you could tell me a little bit about that and what it addressed and your participation in that.
So SNMMI is actually the most important conference in the field of nuclear medicine. It's a place to be if we have something really exciting about radiotheranostics to present. And that's why I'm really curious to hear from Dani what molecular partners had to share with us there.
Yeah, that's what I was already alluding to, is like at SNMMI, we presented the first update, basically the preclinical data package of our 212 lead DLL3 targeted radio-DARPin. So maybe quickly trying to dissect the molecule here and give you a bit of background. DLL3 is a target that is very highly expressed in small cell lung cancer, so a high prevalence. And there is, of course, now other DLL3 moieties like tarlatamab are moving forward. This is a T-cell engager which delivers great benefit to the patients, but at the same time, you see there is only a 40% response rate. So there's a lot of patients that still have a high medical need. And you can imagine, especially those patients where you don't have T cells or no functional T cells in the tumor, they will not respond. So there is clear medical need for other DLL3 targeting or targeting reagents with other payloads to deliver greater benefit to these patients. And that's what we've been aiming for, generating a DLL3 specific DARPin with all the features I explained you before, like the surface engineering, stealth engineering, and the half-life extension, and hooking up these up to lead-212, which is a alpha emitting or a precursor of an alpha emitting particle, which has shown, and we're doing that together with Orano Med, and the lead-212 has shown very great success in the clinics. With first data with phase 2 data that has been presented at ASCO, strong clinical data in terms of efficacy and safety. So that's basically been our starting ground, and we've presented now at SNMMI the data showing on one side good safety, high potency of the molecules, comparing also to benchmark antibodies like the antibody derived from ROBA-T, and of course a strong biodistribution profile speaking for moving this molecule into clinical development, reaching up to 60% injected dose on the tumor at 24 hours. So really strong data package that convinced us and many experts like Ken to move forward with that molecule into the clinics.
There was so many talks about PSMA, SSTR2, and not many of new targets. And that's what I heard. You're standing out. You're going after other targets. And obviously, as Dani said, DLL3 is just the start of it. And we are not bound to the classical targets. And this is the reason we went into this field, was to work on targets others will not. And that you could see at SNMMI. This was one of the few talks that was not on the classical targets.
My question is, when you talk about DLL3, it's very difficult to ignore tarlatamab. I would like to hear your position on tarlatamab. Is this a good thing, a bad thing?
Because molecular partners, we also have T-cell engagers. So I'm not speaking against Amgen. I just speak out of our own experience here with T-cell engagers. And I mean, the whole field of immune oncology is great. The idea to harness the patient's immune system to kill the tumor is great. And in some cases, that means cure. Now, the problem is, and we have our own trials, you are very at the mercy of the immune system of the patient. As Dani said, are the T cells there? Is the tumor what we would say hot? Are these T cells activation? Can you activate them? Or are they exhausted? And to that point, you don't control it. And what I personally like a lot about the radio space is that it is sort of the other side. Radioactivity is a proven modality. Targeting is a proven modality. And together, we don't expect the dependence on the patient's immune system for those patients that need it. And that's the beauty here. This is known that actually radiation can boost the immune system. So there might be a space in the future where we use radiotherapy to get the immune system running and then come in with T-cell engagers, other IO drugs to use the activated immune cells that we have generated with radiotherapy.
And would you say that the approval of tarlatamab is kind of even validating now the target of DLL3?
Totally.
And making for us even more interesting to follow?
Absolutely. When we started working on DLL3, it was an unvalidated target. And one should also say maybe one other thing. So DLL3 is actually a protein that usually is not on the cell surface. So it is not found on healthy cells. In these cancer cells, the direction or where this protein is found changes, and suddenly you have an intracellular protein on the surface. Now you have a sort of a source code to look for these cells, and it is very clean. So no other healthy cells have it on their surface. The problem with DLL3 is there is not much there. So it's a nice, call it black and white situation, nothing in healthy cells. Yes, very clean expression, but not much. So for some ADCs or other approaches, you will not be able to deliver enough potency to kill the tumor. You need to go with high potency drugs as T-cell engagers or radiotherapy. And even for radiotherapy, maybe you might want to explain alpha radiation versus beta radiation. And obviously for me, this is an alpha target, but maybe Ken, you can give us some background on alpha versus beta radiation.
It actually brings us back to the high school when we learned about physics and the different particles, obviously, right? And they're different in ways of radioactive decay. And one of them are the alpha. The alpha have a high energy. They have a very short range, but they are really like almost like a sniper. And then you have the betas, which have not so much energy. They travel a little bit longer, and you have a lot of more crossfire effect. And to be honest, the majority of experience we currently have actually is with lutetium-177, which is predominantly a beta minus, a beta emitter. And we do have a little bit of experience with alpha emitters. I mentioned previously radium-223, radium, or so-called Sofigo. But of course, now we have a lot of hope that in this new generation of radioligand therapy, we are using alphas, which have a very high energy, which very focusedly deliver the radiation to the cells, which express the target and do not affect so much other healthy tissues. The second difference is that you have double strand breaks for alpha interaction. For beta evaluation, you usually have only single strand breaks, which means we hope that there's less recovery, less repair post-alpha. And this is obviously something where we also hope that not only the delivery is more like a very precise sniper way, but also that once we hit a certain cell, that we also really destroy it and make sure it doesn't recover.
For us, of course, an important dimension was to pick the radioisotope that matches to, on one side, the indication, small cell lung cancer, DLL3 as a target, and also the profile of our vector. And that's where we started the collaboration with Orano Med, and Orano Med, who is a leader in the target alpha therapy field using lead-212. And lead-212, for me, has few specific, I say, scientific advantages why we really would like to work with that radioisotope. So first, it has a very short decay half-life. That means you have a lot of energy deposition in a very short time frame on the tumor. And it has a second aspect, which is not yet fully explored in that sense. It gives time for the immune cells afterwards to come in and start to do their job in the tumor. So the radioactivity is not there anymore, has been decaying, and then the immune cells can start to do their job. The second thing is, like, from a safety perspective, it has a very clean decay chain in the circulation. So if it decays in the bloodstream, there is no radioactive daughter radionuclides, that's how we call it, release that can have damaging aspects to other parts of the body, and also the waste management. So especially urine that is excreted by the patient does not need to be collected and stored for a long time. And this is just nuances in the whole aspect compared to other radionuclides, why we like the lead-212. And on the other side, of course, Orano Med is a collaboration partner, extremely strong team, dedicated and strong team, and great complementarity in terms of skills, in terms of having endless supply of, I would call almost endless supply of lead-212 in that sense, having great capabilities of running preclinical work as well as clinical work and supplying the radioactive nuclide for preclinical and then clinical work. So I see a lot of benefit in the collaboration, but also using lead-212 as a radionuclide.
I will echo what Dani said on maybe, or give a bit more background on Orano Med. So Orano Med is the daughter company of Orano, which is the French nuclear power company. And they've been obviously working in the field of nuclear power since a very long time, and also have an investment arm to invest in the next types of energy. And they thought, I guess, let's see what else can be done with nuclear. And that's where they realized that the medical industry, they have an advantage. They have invested heavily in Orano Med. And I think you need both. You need the deep understanding how to handle radioactivity and not be afraid of it, and actually then find out how you do this. And that's where all the knowledge of Orano meets Orano Med, which is then translating this into a therapeutic advantage. And really kudos to my team for finding Orano Med, because this is a private French company. It was not that obvious in the beginning. And luckily we struck that collaboration with this quasi-unlimited supply, really dedicated team and deep pockets for those investments, because they have invested hundreds of millions in the supply chain. So that will not be us, that will be our partner Orano Med that works on the supply chain to solve that. And I think there we can really, it's great to have a trusted partner.
And if you follow the news, and there's not only this fantastic production site in Indiana, where they together as FedEx try to ship out and serve the whole US, but they're also, I think, building a production site now in Europe, in France, if I'm not mistaken. And this will, of course, also enable you to cover the market in both directions.
Where are you at currently with clinical trials? And is small cell lung cancer your key indication?
So obviously, small cell lung cancer will be the first, and where we want to go, but also the target DLL3 is also found on some neuroendocrine tumors that we will also look at. So wherever DLL3 is expressed, we would like to go after it. That will be our first in human trial, and we aim to start clinical trials. So today we're preclinical, next year. So next year we will start to collect first clinical data. On the one side, that will be exciting, but I also heard you, Ken, talking earlier, colorectal, breast cancer. What else did you put out there? So there's lung cancer. So it's naive to think that one target, one drug will be kind of the solution. We will have to go after a multitude of targets, and that's what we're actually doing. So in our pipeline, I think we have roughly 6 targets on the way. And we're always thinking, again, which are those tumors that are radiosensitive, which are those indications where we have the best acceptance of the doctors to go forward. That's why it's great to be working with you, Ken, and get that information. And where can we generate the data that then also excites the patients to sign up for this type of therapy? And I would say we have a good list of targets for tumors. And we're also thinking about even the next step, because our background is multispecifics. And we can do 2 binding sites in one DARPin, an advantage that will be used going forward for our pipeline. So also thinking about targeting different cell types in a tumor. Sometimes it may be a stromal target, a tumor target. So really opening that space for a next generation approach. And very excited to be pushing forward on the clinical trial, but then broadening the pipeline.
A lot of innovation right now is in the target, right? But you cannot protect the target. So technically, once you have disclosed a certain target, a new target, pretty much everyone else can follow. How I see it, but please correct me, the beauty of Molecular Partners is that the DARPin platform as a technology platform is something which is not so easy to copy and paste. Whereas, for example, an antibody, more or less everybody can try to develop an antibody against a certain target.
First of all, the DARPin platform, as it did exist 3 years ago, it was not suitable for radiotherapy because of the kidney and uptake and the lower tumor targeting or accumulation. So only Dani's team has brought this now to the level of that we can say we have a new platform. And this platform with the tricks, we have obviously secured via IP. And maybe, Dani, you can speak a bit. I mean, it's still a lot of work. It is far from trivial to make these DARPins. So we are in the middle, I would say, of a learning curve here.
All makes sense what you said. And it's really like we're in the middle of a learning curve. I think DLL3, that's the showcase. We can do it. We've proven now we can do it. We can generate those molecules. And DLL3 is almost like a bit of a special case in that sense, because we picked a target that is like biologically makes a lot of sense, but I believe others can go for it as well. What we're focusing now in the pipeline of molecules to come is like, on one side, way more tricky targets to address for other modalities, so challenging targets in the sense of how you need to bind them to have the desired specificity for the tumor, and other aspects where we believe the DARPin brings in a uniqueness. So always focusing on where can we build something that is pretty unique. And on the other side, as Ken and Patrick mentioned, it's like the multi-specific space. That's basically our home turf, where we've been now for the last 15 years working on multi-specific therapeutic molecules. And now with the learnings we've made in the radioligand space, building that platform for DARPins, now we start to combine these 2 platforms. So doing multi-specific radioligand therapeutics, as Patrick mentioned, for example, you say I pick on one side a tumor target and I integrate into the same molecule a stromal target. So I hit 2 parts of the tumor with the same molecule. So there's a lot of permutations and thinking of what is the underlying tumor biology we need to address, how can we deliver the radioactivity to a high amount, but also very homogeneously to the tumor to have the best possible effect for patients.
What do you see as the current trends?
I mean, there are many pathways, right? So first of all, we will see the currently approved therapies hopefully see moving to earlier lines. Give you an example, prostate cancer, we started very late in administration, castration-resistant prostate cancer. We now have a couple of studies in the more sensitive setting. There are a couple of IITs even prior to initial resection of the prostate. I think this is one of the trends. The second trend is, of course, combination therapy. I think none of our therapies are curative outside of thyroid, to be honest. And this is something we are going to work on in the future. Next thing, of course, very important is we try to better understand how to personalize medicine, to really treat not all patients the same way. And the fourth one, I think this is the most exciting one. We want to find new target structures. And because we can really image very quickly the target expression, we can also image very quickly the way such new therapies work, that hopefully we establish together with the industry, with partners like you, biotechs, but also with established pharmaceutical companies, so-called fast-track mechanisms to bring these drugs as soon as possible into humans, and not do the typical 10-year program, killing 98 of 99 programs, but really trying to together jumpstart this development and hopefully really tackle these big solid tumors in the future together.
You were mentioning the clinical trials earlier. What's the mode of delivery to the patient?
The dominant way of injection is intravenously, so very similar, let's say, to intravenous chemotherapy. The injection itself is somewhere between 5 to 20 minutes. It's not very long. It's true that the other activity, which is not binding to the tumor, is predominantly excreted via the kidney and the bladders. So it makes a certain sense to collect the urine if possible. Regarding the pathway and how this is happening, it's a little bit heterogeneous in Europe and the US. So you have, let's say, very liberal regulations, like, for example, in the US and also more recently in Italy, where patients can be treated in the so-called day hospital and after 2 or 3 hours leave immediately. Or if you go into these radiophobic countries, including Germany, where patients have to stay 2 nights in the hospital. Overall, I think this is one of the factors people very often underestimate is it's a very well-tolerated treatment. When you look at the success of the current treatments, it's not only because they perform better than established chemotherapies, but they also significantly improve quality of life of patients. Again, one thing is important, the whole administration process. You don't feel much about it, it's not painful, you don't lose your hair. And the second thing is overall, it does not only improve the survival of patients, but also significantly improves the quality of life. And it is something which I also had to appreciate over the last years, talking more and more with patient representatives is, yes, they want to live longer, but even more than living longer is they want to live well. And this is still something when we talk about clinical trials, we sometimes underestimate how important quality of life parameters really are for success of such a therapy.
What's the frequency of treatment? How often do they need to take this?
So this is an excellent question. So right now, we do have therapies where we perform the treatment every 6 to 8 weeks, somewhere between 4 to 6 cycles. But we also see a certain amount of innovation there. There's, for example, in the prostate space, currently a prospective phase 3 study ongoing, where they try to reduce the treatment to only 2 applications. And there's another one, very interesting. It's a complete different program where they actually treat the patient only once, and then they wait until the patient progresses again, and they retreat it. The last one is the HK2 target, and the program is by Janssen, and it's going to go into phase 3. Very, very innovative trial design. The other one I mentioned, the phase 3 study prospectively, is by Telix, where they only apply 2 cycles. So there we see a lot of research and work also done on this, but right now it's 4 to 6 cycles.
To wrap things up, maybe you could give your impressions of where you see the radiopharma field developing.
We have opened the door for radiotherapy, especially in prostate productive drugs. With the DARPin platform, we have now established a novel therapeutic modality in that space that opens the door for more targets, for more indications, and also likely for combinations of targets. For that to work, we needed to access the lead or the alpha supply via Orano Med. Also, that box is ticked, and we're very confident that they will work and solve the supply chain questions. And for me, the vision, and that's what actually caught my eye in the beginning of the whole field, is this idea of you see what you treat with imaging and you treat what you see. To Ken's point, I mean, it's the first time where you can actually literally monitor the tumor and then treat. And I think this in today's life, when we want to see things, understand things, this is really the promise. And going to more personalized, more drugs, and even those combinations, that's where I see this field going.
So for us, as a field, it's a very critical phase. People are aware of us to some degree. Now we really need to, first of all, make sure that the therapies we have available, we bring to patients. So we really need to handle the scale up. The second thing is we really also need to make people aware of us. So that's why I'm very grateful for this podcast, giving the opportunity to make people aware what radiotheranostics is. Everyone knows what ADCs is. Everyone knows what chemotherapy is. We still don't do this enough. And the third one, the most important, is to make this growth sustainable. We need to come with new therapies. We need to go beyond somatostatin 2 receptors, PSMA. We really need to tackle other very important solid tumors, very other important solid targets. And this is something which we can only do together. And that's why I'm very grateful that the Molecular Partners gang invited me to this podcast, because we need to work together, academia, patients, and industry. So I think this is a very critical moment. But if we continue to deliver, then we are here to stay.
A really interesting subject and a great conversation there. Definitely a field to keep an eye on in the future. Don't forget to check out the latest news and articles over at Labiotech.eu, and I hope wherever in the world you are, you'll have a great week ahead. Thanks for listening, and I hope you'll join us next time for another Beyond Biotech.